Good morning, everyone, and welcome to this masterclass named Biology to Biotechnology, an Overview, organized by SaiU School of Technology. It is my absolute privilege to introduce the speaker for today, Professor Subba Rao, the Dean, School of Technology at Sai University. Professor Rao brings with him over 34 years of scientific leadership from Baba Atomic Research Center, Kalpakam, where he led pioneering work in basic, applied, and molecular microbiology. A fellow of the Academy of Sciences, Chennai, and the Society for Applied Biotechnology, he has made profound contributions to field ranging from biocorrosion and biofolding of wastewater treatment and environmental impact assessment. Professor Rao earned his DSc in biotechnology and his PhD in marine biotechnology, and his academic journey has also included a postdoctoral fellowship in Germany and advanced studies in microbiology, biochemistry, and immunology as well. He has published over 250 peer-reviewed papers, executed projects worth 12.6 crores, and was recognized among the top 2% of the world's most influential researchers. Beyond research, Professor Rao has mentored numerous PhD and postgraduate scholars and actively contributes to national and international scientific boards and editorial panels. With that, I welcome Professor Subba Rao, sir, and over to you. And quick announcement, participants, the questions will be at the end of the session. So you can post in the Q&A section or in the chat box, and sir and ma'am also will answer that. Over to you, sir. Thank you.
Yeah. Thank you, Nellai, for the introduction. Welcome, dear participants. It's my pleasure and privilege to talk to you about the scopes of biology to biotechnology. So in this half an hour or so presentation, I would like to share the history as well as the developments and what are the current activities that have been happening in the area of biotechnology. Yeah. So I have structured the talk in such a way that this webinar is an engagement for you students towards the process of discovery and in exploring topics at the forefront of mankind, and to inculcate or reinforce a scientific perspective with education. That is what I would like to do at the School of Technology in the context of the courses like BTech in Biotechnology, which we are going to begin from this academic year. So now coming to a degree in biology or biotechnology, particularly in the context of liberal education or liberal arts, actually prepares students not only to make a living, but also a life. So this is what actually I feel the benefits of studying at Sai University in the context of your choice of undergraduate degree, as well as the plethora of various courses which are offered to you as part of the liberal education model. So if you look at liberal arts and sciences, they have been a foundation for colleges over centuries, actually. And they are also more important and useful today. And it includes a lot of possible subjects and areas of study, which actually builds a professional career for a student. And then you can also explore the study of arts, hard sciences, social sciences, literature, environmental sciences, biotechnology, computer science, maths, whatever you name. And you can combine your interests and talents into a unique education of your choice. Actually, this is what is that Sai University is going to offer to you in the context of your educational career at the undergraduate level. So coming to why this liberal education and then science and technology, what are the options, and then what is that it is going to give you is that you will learn the required skills and how to think and communicate. Actually, this is as part of the interdisciplinary core courses which we are offering for the students in the first 2 semesters of their undergraduate degree, is to experience you yourself and also understand the need of accomplishing a life and lay a solid foundation for higher studies actually. This is the one of the major objectives on which uh, you can know your passion and also know yourself better once you look into a undergraduate education journey, and it will give you almost experience and also a commitment for a lifelong learning process. And you have also a flexible degree that appears to an employer, and any employer can train easily on job specifics if you have such kind of uh, experiences in your undergraduate education. Also, this kind of study will teach you the ability to communicate ideas, analyze, and then also look into the information and data, and as also work in teams and also solve problems together. So if you look at across the subject areas, education at SaiU teaches what today employer value most. Actually, they look at your communicating skills and also in your ideas, also the analyzing information and data, and then adapting to new technologies and professional trends. So now this brief outline of your undergraduate education and then scope and opportunities, I would like to introduce to you the word what is called as biomimetics. That is nothing but you are going to mimic the nature and also use the nature's architecture in building a very strong architectural marvels. Actually, if you look at this La Sagrada Familia, which is a church in Barcelona, Spain. It is built by Anthony Gaudi. So he was inspired by the geometrical features found in nature, like unique branching of tree columns and also staircases with shell-like curves. That is what he has created in this marvelous architecture, and also designed the physical structure of the church in such a way that even today people can appreciate the kind of architectural infrastructure which was built into this. So this is nothing but if you mimic nature, that is particularly the biological aspects, you can able to bring in wonderful architectural marvels in structures. Likewise, if you go to any beach and you can see these kind of shells, and then these kind of shells will have this kind of patterns. These patterns can be molded onto a micro structural polymeric matrix. And these are the ones actually which will prevent the settlement of any organism on their surfaces. Likewise, we have also looked into the application of these biomimetics in the context of preparing polymer coatings, which will protect the ships and then also protect the cooling water systems which are present in most of the industrial cooling systems. Now, another interesting area where actually the importance of understanding the complexity of data and structures is the connectivity of the brain. Actually, it is where that Daniel Lee, a neuroscientist, viewed a multidimensional network in an artistic lens by enlisting artists and designers to help visualizing brain connectivities. So this is where actually the inspiration has enabled cutting edge technologies such as virtual reality and rapid prototyping, which have been developed and which are becoming increasingly accessible and inexpensive techniques in the context of biomedical research. So now that you may be hearing that a lot of research activities are being centered towards almost understanding or mapping the brain. So this is where actually the major medical research activities are happening, particularly in the context of neuroscience as well as and also in cancer biology. So now if you look at the biological sciences as a fundamental aspects of towards any applied or technological development, yeah, you can encompass all the divisions of natural sciences in various aspects of vital processes, which will play a major role in the life processes of any organism. So the concepts include anatomy, physiology, cell biology, biochemistry, biophysics, animals, and plants also. So biological studies are on Earth, from the simplest to the smallest single cell organism to the complexity of human brain, have almost excited the mankind. And the science of biology shapes everything from agriculture to zoology. So that is from A to Z. So if you look at this schematic illustration, starting from atoms, which will range from Armstrongs to nanometers to microns, the simplest life form, the viruses, will be in the range of about 100 nanometers. And then look at the frog's egg, it will be just 1 millimeter. And then an ant will be generally around 1 centimeter or even less than that. There are few ants which are even a millimeter level. Then coming to humans, about 1 meter. And then the blue whale is the largest, that is more than 10 meters. So if you look at from 1 millimeter to close to about 100 meters or 1 kilometer, the human eye has the capabilities. The rest from 1 micron to close to few hundred microns, you have to use a light microscope. And you need an electron microscope if you would like to see from nanometers to close to 10 microns or 100 microns. So now looking at the life science processes, it all starts with a molecule, which is the biochemical constituent of a cell, which on joining together molecules can develop into organelles, which is a specialized subunit in a cell. And then you have the cell, which is a functional unit of a living organism. And a cell of group of things set to interact together are called tissues. And the tissues will result into organ development, which has a functional activity. And the organ system, which is a specialized functional system of a living organism, will result into a single individual organism with all life functions. And a group of organisms will result into population, and then the population groups will result into community, and the community interacting with the environment, abiotic environment, will result into an ecosystem, and you have finally the biosphere, which is the world we live in. So now we look at some of the biological discoveries that have revolutionized the life sciences, include the Darwin's theory of evolution, which is nothing but the natural selection, which he has published in 1859. So Darwin describes how organisms evolve over generations through the inheritance of physical or behavioral traits. And then you have the wonderful discovery of penicillin by Alexander Fleming, which has almost revolutionized the medical and the health research. And then you have the next major development is the HeLa cells. So HeLa cells are immortal cell lines that are of immense value in scientific research. They are most commonly used human cell line, and this was derived from the cervical cancer cells taken from Henrietta Lacks, a patient who died of cancer, cervical cancer, in 1951. So these cells are the one which are giving us exactly the human tissue outside of a human body. So that's where many scientific and also science adjacent fields took advantage of these cell lines to study a variety of things from disease prevention to space physiology. So this is one which has revolutionized completely the scientific area of research in medicine and healthcare. Then major discovery was the DNA structure by Watson and Crick. And this, as you know, has resulted in the development of molecular biology, recombinant DNA technology, and many, many other things. And then you have what is called as transformation, that is the gene transfer, which has almost revolutionized the incorporation and also into the development of recombinant DNA technologies. And then you have PCR, the polymerase chain reaction, which is fast and inexpensive technique, which is to amplify a copy of small segments of DNA. And all of you might have heard about its importance as part of the recent uh, global outbreak, the COVID, where all of us were subjected to a PCR test to confirm whether we have the COVID virus or not. So it is Carrie B. Mullins, who was awarded the Nobel Prize for Chemistry in 1923, who has discovered this polymerase chain reaction. And then you have bioluminescence markers, the green fluorescent protein, which was extracted from the jellyfishes. And this also has resulted into revolutionizing the biological research and has developed into many biotechnological processes and identification kits. And finally, many other discoveries of this fluorescent protein markers has resulted into a variety of applications in both cancer biology and also in biotechnological areas. Now, another interesting discovery was the dolly, which is nothing but a cloned female sheep using the adult somatic cells from the mammary glands. And then in the process of nuclear transfer, they are resulted into this small offspring, dolly. And dolly was a significant biological breakthrough. And based on this, now other animals have also been cloned, like pigs, deers, horses, and bulls. And then people are also experimenting on the extinct animals and resurrecting them from the frozen tissue. The woolly mammoth is the one which people have almost completed the research and would be able to expect the results soon. And then we have the stem cell creation from the skin cells. So it was in 2007 that the skin cells were reverted to a pluripotent stem cell property. And this new process of creating pluripotent stem cells has changed the programming of the human cell and also resulted into developing a variety of stem cells like kidneys, liver, heart, lungs, brain, etc. And now this is has resulted into a stem cell therapy, which is almost revolutionizing the healthcare in the context of treating many dreaded diseases. And then the major significant development is the genomics and proteomics. So genomics is nothing but the study of organisms and then their genome and the use of the genes and then understanding them. Generally deals with the systematic use of this genomic information and then to provide answers in biology, medicine, and also in the biotech industry. And then proteomics is the large scale production or study of proteins, particularly their structure and function. So proteomics and genomics have almost revolutionized the biotech area. And these are the ones which are giving money spinning new healthcare products as well as pharmaceuticals, which is showing quite a promise and then also great opportunities for biotech startups or biotech industrial opportunities. Then you have what is called as the identification of organisms. Generally, we use the 16S rRNA gene in the context of bacterial identification or characterization, and then 18S rRNA for the eukaryotes. And in the context of bigger organisms, we are using cytochrome oxidase 1. And then these are the ones which will give us the barcoding of the particular genomic aspects of an organism. And then the sequencing technology, the Illumina sequencer is the one which is the most advanced, which will provide you sequencing of billions of genomic genomes in a very few days. So this high throughput meta barcoding will enhance the precision of species detection and also identifying the biological community. So if we look into the classical taxonomic feature, that is almost, people are forgetting about it, and also we have almost lost most of the things and also the expertise. Now it is only the barcoding aspects which are now identifying the biological aspects of any organism. Then the area other which is catching up is bioinformatics, which is a subdiscipline of biology and computer science. And then the applications include determining the gene and the protein function, and also establishing evolutionary relationships, predicting the 3D shapes of proteins, or many more. So how can we measure metabolic processes and gene activity? This is where the informatics or the molecular informatics is going to play a major role. And then the modern genome researchers have mapped the entire human genome, that all of you already know about it. And then we know that entire 3.3 billion base pairs of the human genome, but what it does and what it means, it is still not yet cracked. There is a lot of scope and opportunity in this context. So another area is computational biology, where biology and mathematics have intertwined, and the dynamic interplay of these hosts and parasites, molecular forces in protein, biological patterns, signal transmission along the axons of the brain cells, and then using the tools of mathematical analysis, such as the non-linear dynamics, and then partial differential equations. People are developing a variety of algorithms to understand these biological processes and also attaining them to a technological application. So biology became an information-based field as of now, and it is also growing at a rapid pace, that the algorithms from the statistics and the machine learning are applied to genome studies and also for classification. So if you look at these illustrations, this particular thing is about the structure of biomolecule. Machine learning is transforming our understanding of this protein folding mechanism based upon this variety of protein folding processes, and then also transforming or modifying these structures using computational algorithms. People are developing new highly effective drug molecules or nutraceuticals or pharmaceuticals. And then next is the fluxomics, that is to understand the flux of metabolic rates at which the reactions of biological entity are happening. You may be surprised to know that femtosecond, picosecond reactions are being monitored in biological processes now using sophisticated laser scanning spectroscopy equipment. Likewise, with the advent of artificial intelligence and machine learning, the hypothesis testing and knowledge discovery of anything is happening in a better way now, based upon what is called as in silico analysis. That is, without doing any experimental studies, you can able to prepare an hypothesis based on the biological and physiological knowledge and data available, and then develop models or algorithms, and then make a dry experimental run using computational power. And then these will result into a very efficient, simple, and finalized experimental designs to test your hypothesis. And then once you do the wet experiments and verify your hypothesis, Either you can reject or accept it. So this is how the experimental technology also has been developed in the context of molecular biology, genomics, proteomics, or metabolomics, along with artificial intelligence and machine learning. So the advent of cryo electron tomography, which offers looking at cellular landscapes at a resolution of less than 10 angstroms. So you can also predict to attain atomic resolve in the context of biomolecular processes. And in addition, you have microfluidics and microfabrication approaches, which are generating the ability to monitor cells and their components at unprecedented levels and resolution, actually. Likewise, you might have heard about 3D printing. Now, 3D printing of organs is been happening. So, in the context of any major accidents where you are able to lose many of your facial structures, these are all being printed, tailor-made exactly to your body, and then they're fixed. They also have printed a 3D heart and also the cranium structures which are damaged in a major accidental process. So 3D bioprinting is an emerging engineering field which has wide medical applications, and then doctors, patients, and then the communication. So this is how the interoperative navigation, experiments, pre-operative planning, stimulating operations, medical education is all getting transformed in the context of this 3D printing biotechnological processes. Likewise, tissues are also being printed now. And another development is the 4D printing. It is a process through which a 3D printed object transforms itself into another structure over the influence of external factors such as temperature and others. And then 5D printing is a new branch of additive manufacturing where the printing head and the printable object have 5 degrees of freedom. So these are also revolutionizing the tissue engineering research, biosensor development, bioactuators, and biorobotics. So biorobotics is the one which is almost advanced research applications. So another recent development is the synthetic biology, which is nothing but you will be using the engineering of a biological system, and then is to develop a biological module as the components with which one can engineer a new biological system. And this hypothetically leads to cells and then organisms or biologically inspired systems with industrial or therapeutic application, actually. That means you will be using exactly what is the required genomic structure to be cloned into a system, and then you can have the products from the cell, such for example proteins, and these are the ones which you can downstream processing becomes quite easy because you'll be having only this particular single protein which is going to be uh, delivered by the cells which using a synthetic biology process. So it applications molecular biology, bioinformatics, computer science, physics, engineering science, chemistry, agriculture, and microbiology. So this is where now actually the transdisciplinary interaction, which is the one which is going to revolutionize the biotech research. Likewise, in the context of wastewater treatment, now we have developed bioreactors where you can do all the process of filling, mixing, reaction, settlement, and drawing of the end product in a single tank. This is how a single microbial granule looks like. And these are the granules, which the size is about close to 2 millimeters. They can be stored for about 6 months in a shelf without any preservatives. And this is where actually we have applied this for the wastewater as well as in the sewage water treatment. So now various technologies have been developed. These are the granules which have been used for a variety of other organic molecule degradations as well as in the degradation of sewage and other industrial waste products. So based upon the scale-up studies, a full-scale project are functional at the Kalpakam township, which about 1.5 million liters per day plants have been uh, designed and then also being in operation now. So this shows that you can have a very small footprint, and it also uh, reduces the cost and also energy needs as part of the conventional activated bioprocesses in treating of wastewater. And also, apart from those granules which are based upon bacteria and fungi, we have what is called as phototrophic granules, which will be developed into different sizes. You can design them as per the application part. You can see this image where the raw sewage is being treated using these granules, and you can see the final clear water which is coming, which is directly can be used for gardening, and if it is further treated, can be used for flushing toilets and all. So thereby you can reduce the uh, wastewater released into the environment. So this is the scale up of the photobioreactor, which is quite simple. You can directly use them in the sunlight, open sunlight, where they capture carbon dioxide and then sunlight and then get the energy. There is no need to provide any other organic source for the growth of this kind of phototrophic granules, which are nothing but microalgae. And you also have wonders like you may be aware that gray is the one of the unit of radiation, ionizing radiation. 6 gray is enough to kill a human. So 15,000 gray is the radiation resistance of this bacteria called Deinococcus, which is one of my most major researched organism as a part of radiation waste treatment. So this is how this organism grows, and then these are the images which are recorded as part of their growth. And this is the one which is having a tetracoccal structure, and then these are the ones which are having the green fluorescent protein. You can directly see using a scanning laser scanning microscope or a fluorescence without even staining anything. It has wide applications now in context of radiation waste treatment. Likewise, environment impact assessment is another area of emerging environmental biotechnology. So here, industry uses water, seawater through this dike. Example is this is the Kudankulam nuclear power plant, and then the water goes inside, cools the condensers, the heated water is released. So we also should know how this heated water is going to get dispersed in the open sea. So these are the uh, images which have been developed using the geoinformatic software system and mapped the dispersion of the heated water. Heated water will be just only 4 to 5 degrees above the ambient water of the sea. So this is based upon different months, the dispersion based upon the sea current movement that we have recorded. So it also has a wide application in the context of marine productivity. Another is the deep sea studies, wherein the Sai University has started research in the context of understanding the deep sea biodiversity and also understanding the deep sea biological processes. So if you look at the deep sea zones, you have the continental shelf, and then you have the bathyal zone, abysmal zone, and then the deep sea trenches, which are as deep as 11,000 meters. So we are part of this national mission on deep ocean mission, and then we are able to do some basic studies in understanding and isolating the biodiversity which is present in our deep seas, the both east and west coast and down in the Indian Ocean. Another aspect is the deep sea mining, where the manganese nodules or other uh, strategic metal have been extracted using this deep sea mining technology. And another significant issue is the ocean acidification. All of you maybe know that the carbon dioxide levels are being increased. Now they are more than 400 ppm in our atmosphere. This is resulting in the context of global warming. And then as a result, the sea level rise, accelerating melting of the ice at the polars, at the poles, and then the warmer atmosphere is resulting in more water vapor evaporation and increased heavy rainfalls and flooding. That is what we are seeing as of now. Likewise, the ocean acidification is also impacting the chemistry of the shells, which are formed into nothing but calcium carbonate shell. And ocean acidification, now don't think ocean becoming acid like thing. No, just a drop of 0.1 or zero point 0.2 units of the pH is resulting in insignificant calcium carbonate shell formation actually. So now look at the current developments in biotechnology. You have what is called as gene editing through the CRISPR technology. CRISPR is an acronym for clustered regularly interspaced short palindromic repeats. And this has almost is revolutionizing the uh, DNA technology. And then also bioinformatics is the one which is playing an important role. And then bioprinting, as I already mentioned to you, in creating functional tissues and organs synthetically. And green biotechnology is the one which is able to develop sustainable solutions for our agriculture and energy and environmental conservation. Artificial intelligence, machine learning are also playing a big role in the context of diagnostics and personalized medicine research and development. The career opportunities for biotech are in the research as a research scientist, working in many academic or research organizations, as a biomedical engineer in many healthcare establishments, and in clinical research. And then you have quality control analysts, and then bioinformaticians. As an entrepreneur, you can start and manage your own biotech company. And then in sales and marketing of biotech products, intellectual property research and patenting processes. We are also in legal advisory for biotechnology. And finally, in teaching or sharing knowledge and expertise in biotechnology. This is what I would like to share in the context of how from biology we got transformed to biotechnology and then its applications in the current mankind applications. Thank you.
Thank you so much, sir. So, participants, with that, so it's time for Q&A. So, if you have any questions related to the topic or related to the programs, the BTech Biotech at Sai University, you can very well post in the chat box. So, sir will be happy to answer that. We also have Manobala ma'am, so she'll also be able to. Uh, if you can't find the chat box, you can find it in Q&A, and you can very well post the questions over there. You can also raise your hand. We'll enable you to talk. So, any questions, participants? So we have a question from Rudra Ishwaran.
Yeah.
How do you view the use of vectors in gene therapy? Will there be synthetic vectora eventually?
Can you repeat the question, please?
So how do you view the use of vectors in gene
Uh,
therapy? Will there be
yeah.
synthetic vectora eventually?
See, vectors are the ones which will be the vehicles for the gene to be cloned into a particular organism, actually. So, if you want to particularly express a particular gene, you have to choose the right vector and then also design it in such a way that it has its expression like it should be overexpressed. And this is how finally, when you are, what is the protein or a particular product of your interest is produced in significant concentrations for applications.
Thank you, sir. Participants, do you have any more questions about the program or about the topic? You can very well post in the Q&A, or you can raise your hand. Yeah, Rudra. Okay. Yeah, you can now. Yeah.
Sir, just a follow-up question, sir. So, from my understanding, sir, in the modern day, usually they use the use of vectors comes from certain kinds of retroviruses and related viruses, right, sir? So, the question I have is that if we take antigens of specific cells, right, sir, is there a possibility, in your opinion? I'm just curious. Would there be a possibility eventually where um, a synthetic like a biotech can be made, a biotechnological vector, a non viral vector can be made to be able to detect surface antigens in order to have a more targeted effect, because viruses have a capacity for like gene damage, right, sir? So that's why I'm curious.
Yeah, Rudra, actually, synthetic biology, which I have mentioned in one of the two slides which I described, that is the one answer to your question. Yes, instead of going for any viral vectors, which were quite widely applied in the recombinant DNA technology, yes, we can design synthetic vectors and then make our own choice. That's where actually the CRISPR technology is coming up very uh, rapidly into this area of gene transfer or gene editing technology.
Okay, yes, sir. Thank you, sir.
Participants, any more questions? So we have one more. So how can
Yeah.
we pursue biotechnology and what are the fields under it?
See, after your plus two, if you have biology as one of your subjects, and then also mathematics, it will be more advantageous if you have mathematics. Even then, if it is biology, physics, chemistry, yes, you are eligible for joining a BTech Biotech program. And in this biotech program, particularly at Sai University, we have designed in such a way that you are exposed to all aspects of the technological developments which are happening. We have also designed in such a way the syllabus that we are providing you more than 20% of the GATE syllabus, which is prescribed for the MTech program to join into the IITs. So that way, the syllabus is well framed and also gives an edge compared to all other universities' syllabi, which we have looked into before carefully designing the syllabus of biotechnology at Sai University. So this is the one advantage which we are going to provide to the students. And also we have two well-established biolabs at the Sai University with all the sophisticated experiment equipment to involve the students into encouraging for the undergraduate research as part of their undergraduate degree process. And this will actually enable the students to understand better when compared to the other conventional universities, which just give more emphasis on the theoretical aspect rather than onto the practical experience for the students. So this is where we are giving an edge to the students who are going to do a BTech Biotech program at Sai University.
బొంబాయిలో అంతే బొంబాయిలో అంతే బొంబాయిలో అంతే
Participants, final call for questions. We are nearing the end of the session. So if you have any questions, you can put in the chat box or in the Q&A.
And even they can put that, and later on also we can reply to them on email also in case they respond little lately also.
Sure, sure. So, participants, as sir said, if you want to connect with our faculty regarding any particular personal questions regarding the program or the topic, can very well reach out to the counselor and they will, get in touch with the faculty directly over an online meeting. So you can very well ask the questions. If there are no more questions, we can end the session. Sure.
Thank you,
Thank you
Nandai.
so much, sir. Thank you so much. Sir, one last question.
Yeah.
A small brief on microbiology. Can you give a small brief on microbiology?
Yeah. Yeah, well, Diksha, microbiology is the one which has actually resulted into all these recombinant DNA, molecular biology, or biotech technological processes. So, in microbiology, you will be learning about the different aspects of or the classification of microorganisms and their metabolic processes, and then also their genomic responses, as the result of which some of them are harmful in causing the disease, so we call them as pathogenic bacteria or pathogenic microorganisms. And there are some beneficial organisms which are used in industries for development of various industrial products, like you may be hearing about wine, beer production, and also in variety of pharmaceuticals. Most of the antibiotics are produced by streptomycin species, and then also many vitamins, and then also many amino acids. A plethora of products which have been provided by the microorganisms. And then, as you know, microorganisms are omnipresent. Even in the space also, they have been identified now. The International Space Center has recorded the presence of microorganisms in the open space, and how they are surviving and how they are thriving with whatever possible organic compounds which are required for that energy are being studied, so that these can be used into future space research, particularly in the context of habitating the Mars planet. Thank you.
Sure, sir. I think we are ending time, so we have 2, 3 more questions. So, if one is, if you can explain quickly on gene therapy, that's the first one.
Yeah.
And second, in biotechnology, there are 2 core types, genetic and biotech. Can you tell about major difference?
So one first on the genetic and biotech part, you see, once we are able to understand the genetic aspects, that is, once you understand the genome only, you can able to transform that genome into an applied technology that is nothing but the biotech. So that's where actually the genomics research or genomic studies will help in developing a biotech process. This happens in the context of any drug design or a new pharmaceutical product or any industrial product or an enzyme or into a medical application, etc. Coming to the uh, gene therapy, yes, there are many applications which have demonstrated using gene therapy now. Like, for example, recently a particular congenial eye disease, which is result of a particular protein production, which results into blindness. The particular gene name, I'm unable to get it, but people have cloned that a particular thing into a stem cell and then injected and then seen that the vision is brought back. So this is how gene therapy is happening. And also in the context of other thalassemia disease and then other sickle cell anemia, the gene therapy is able to provide solution, although it is an hereditary diseases which are carried over generations among families. So there is a lot of promise in gene therapy now. And many areas, even the damaged knees, where people, because of old age, are getting revived using old age, using gene therapy.
Perfect, sir. Thank you so much. I think
Yeah,
that
thank you.
would be end of questions. Thank you, sir.